Good morning. My name is Victor Manjarrez, and welcome to UTEP and the DHS Symposium Series. I think that you'll find the topics spoken about today interesting and very much aligned to what many of you are doing or about to do within CBP and state and local on that. So thank you very much for coming here today. Just a couple of the logistics. There's coffee and tea outside. Please help yourself on that as we go through. Restrooms are down the hallway, past the vending machines, and to the right. There should be some agendas in your folders or your packets that you received on that. So I'd like to tell you a little bit about the Symposium Series before we actually start out with some of the introductions. The Symposium Series is actually funded by Department of Homeland Security, Science and Technology, Office of University Programs, through the Center for Borders, Trade, and Immigration Research out of the University of Houston. On that, so the idea behind the Symposium Series is to provide supplemental training and education insight into topics that are of interest to practitioners. And this is geared towards practitioners. And the topics come up by an advisory board that I've put together. So we meet once a month. It's a Homeland Security Symposium Advisory Board. I have the sector chief here. I have the resident agent in charge from Secret Service, OFO, as well as Commander Marcos Vargas from the Sheriff's Office, Assistant Chief Pete Pasillas from the El Paso PD, membership from the TSA, and a few other folks that helped me decide on some of the topics. Based on their recommendations, I go out and try to seek the people that can deliver that type of symposium material. On that, so like I said, so what you'll see today is a result or a request by your leadership. On that, what you'll see later on is in those packets is an exit survey, and part of that survey in the back asks what are the things you'd like to see or hear about. So I encourage you at the end of the symposium is to fill out those exit surveys, in particular the front and back. The front, we have a couple of questions that we'd like to know how we're doing. In fact, what things that we can change, things that we did well. But the back side's pretty important to us also. That some of the topics that we are planning in the future have come from the comments made in the back. For example, in your packet, there's one for August 10th, and it's the radicalization of domestic gangs. On that actually came out from several comments on the last symposium. We thought that's very unique, so we found the expertise to be able to do that. So we take a look at those. We put a lot of value on that, on those exit surveys, in terms of how we're doing and some of the things that we'd like to do in the future. What I like to do is introduce the three individuals that agreed to join us and present the symposium, and then turn it over to them. On that. First of all, Dr. Jennifer Kovacs is an assistant professor of biology at Spelman College. She earned her PhD in applied biology at Georgia Institute of Technology, and some of her recent work has utilized high throughput DNA sequencing and DNA metabarcoding to identify pollen environmental samples done in collaboration with CBTR. She has CBTR, but actually DHS. She's done some work now, going back a few years with this type of work. Dr. Karen Bell is a postdoctorate fellow at Emory University. She specializes in genetic methods of species identification, also known as DNA barcoding. Her current research involves method development in pollen DNA barcoding for national security forensics. And Ms. Piper Schwenke, she is from the National Oceanic and Atmospheric at NOAA. NOAA at the Northwest Fisheries Science Center in Seattle, Washington. She earned her master's degree from the University of Washington School of Aquatic and Fisheries Sciences. She's also trained and mentored in DNA forensic science at a private crime laboratory in Seattle, Washington, where she worked on evidence from major crime scenes using human identification techniques. As most of you that are here and starting to understand the importance that DNA pollen analysis, things of that nature are starting to play in the jobs you do every day. So with that, I'd like to introduce Dr. Jennifer Kovics.
And I would like to say, just to sort of start the thanks, thank you so much to Victor for organizing all of this. I met him 2 years ago when I came out here to El Paso, and he's truly a force to be reckoned with. And so thank you so much for the invite and allowing me to organize it and come talk today. Leonora as well, just to sort of, she's done a great job, and there's 2 fantastic undergraduate researchers that were there greeting you this morning, Robert and Monica, and so say thanks to them for making these beautiful packets and keeping everything on track for the day. So thank you very much for having me and all of the support that's coming. I think we've organized what I hope is going to be a really interesting symposium. It's been actually a really great process of getting these 3, including myself, researchers together. So we hadn't all met in person until we came here, but we've been meeting virtually and talking about what we do, and we hope that we're going to present something I think is better than what I had thought when I originally had a conversation with Victor. So talking about a lot of different things. It's a really exciting time to do what we do. I'm an evolutionary geneticist by training. I'm an evolutionary biologist. I actually teach at a small liberal arts college, so I have lots of undergraduate researchers. What I do isn't necessarily applied. It's a really basic kind of research. But we're starting to get to the point where we can answer so many great questions with the technology that we have. And the technology is moving really, really fast. So I'm going to provide a little bit of an overview. Karen's going to get into some really nitty-gritty methodology. You can blame her for that test being so hard, I think. And then Piper does a lot of the applications. So she does a lot of applied stuff. She's got some fantastic case studies to talk about some applications of barcoding and sequencing, really just sort of really thinking about how we can harness and use all of this new sequencing technology in an applied way, especially for you guys. So with that, I'll just sort of start. So I realized that a lot of what we do is brand new. It's not in textbooks, right? So none of this was in my biology. We were talking about breakfast. None of this was in my biology textbook when I was a student a million years ago, right? It's pretty fast and it's pretty quick moving. So I'm going to try to get us all up to speed. I'm going to put on my Professor Kovacs hat and get us up to speed thinking about DNA sequencing, DNA barcoding, some of the really basic applications, and hopefully when we have question and answer time, we can hear a lot from you guys as far as what your questions are. So, and what kinds of things you're thinking about. And so I really hope for this to be an interactive symposium today. So thank you very much. And I'm going to go ahead and get started. I'll point out when there starts to get answers to some of that really hard quiz as well. All right, so an introduction to DNA barcoding and applications in DNA sequencing. So just sort of thinking about forensic science in a really broad context. I know a lot of you work in that field and think about these questions all the time. And we can use forensic science to answer questions of provenance. So where does something come from? One of the reasons that I became interested in this and sort of became part of this project, I do a lot of ecology stuff, so I was interested in where the honeybees go sometimes, right? So where do they go? Where do they pollinate? How can we know what they're doing, right? Because they're fast and they're little and they fly pretty far. And one way we could think about that is honey, right? But people have been actually thinking about that for a while. So when we're importing honey, bringing it into the country, is it actually coming from where it says that it's coming from on that label, right? So questions of provenance, is it actually where it's supposed to be? And in fact, several years ago, there was a really big case, some of you are probably familiar with this, of honey laundering, where there was honey coming from China, but we charge a lot more, a much higher tariff for honey coming from China, so they were actually bringing it in through different places, particularly through Germany, and selling it as German honey and not Chinese honey, and this was actually a lot of money, kind of a big case, thinking about that. So how can we identify where something comes from? And we can use we can use forensic science. Forensic science asks those kinds of questions. We can also think about whether a product comes from an endangered species or a population, right? So it's Texas, so I had to put some boots on there somewhere, right? But thinking about are those alligator or snakeskin boots really what they are? There was a case of the fur coats that are kind of up here. They're actually tanuki. So tanuki are raccoon dogs out of Japan. Kind of cute, but they were selling them as fur coats that were not labeled as tanuki fur coats. We can also think about cases of illegal timber and logging. So are these logs from a population that is allowed to log? Is it from a forest where that's actually allowed to happen? And that has to do with the Lacey Act, right? Am I right, Piper? Okay. And Piper's going to talk about that, as well as, are these fish coming from a population where it's legal, where you're allowed to actually fish in those populations? And then, of course, thinking about questions of the ivory trade. So is that thing, that's said to be legally harvested walrus ivory, actually legally harvested walrus ivory, and thinking about that. So these are the types of questions that we're thinking about. And also, in the really broad scale, thinking about, taking a very wide view on this, thinking about geolocation. So where has an object been? Where has a person been sometimes? And what kinds of forensic science tools can we use to answer that question? And I think not surprisingly, today's focus is going to be thinking about, well, how can DNA become part of our toolbox to answer these questions, right? These are questions that have been addressed and been asked and answered without DNA, but how can we start to include DNA in high throughput and next generation sequencing to help us answer these questions? And that's really what I'm going to be talking about today, is a couple of techniques that we use as practitioners in basic science as well as applied science to answer a lot of these questions using DNA, using little bits of clear liquid in test tubes. All right, so DNA barcoding. So there's a couple terms, and I want to make sure that we're all on the same page for them. So a barcode in our parlance, the way we're going to be talking today, is a short section of DNA that's found in all species. So we have several different barcodes, but ideally when you're picking one, you want to have a short section that's conserved. So in animals, we try to have a barcode that works across all animals. So everything from, a cricket in your basement to humans, right? It needs to be in all of those species. But it needs to have some variation between the species. So some differences between species that allow you to identify what that species is. But there shouldn't be too much variation within a species, right? Because if there's a lot of variation, well, then you can't tell where it came from, right? You can't. So it needs to be individual enough, but not too individual, right? So there's been a lot of talk about this. Scientists debate. Well, I think it's pretty much we're still deciding on how to do some of it, but there's been a lot of debate upon which barcodes, what sequences are right, and really a lot of thought has gone into this. And so if you do it right, a DNA barcode should allow you to identify individual species based on that sequence at that DNA locus. So for example, this is a sequence for three different species, animal species, looking at a butterfly and two types of owls. It's the CO1 sequence. It's about 684 base pairs long, right? And you can see that each of those little colored bars, for those of you that haven't seen this before, it looks really pretty, right? But each of those is actually a base pair. And so based on its color, whether it's an A, a T, a C, or a G, right? To go back to your basic DNA class, if you remember that. And you can start to compare them. So if you were actually look at it, most likely our two owls or two birds would look more similar to each other than they would to the moth up there. I think that's a moth. I should know this, but I don't. All right, it's okay though. And you can start to, but each one has its own individual barcode, so its own way of looking. And So this is sort of the barcodes that we use for prokaryotes for bacteria. We use our RNA genes for species identification, though there's not really species in bacteria. That's a whole different conversation. But we use those for identifying bacteria. In animals, birds, and fishes, mostly there's a small region of the mitochondrial CO1 gene. A lot of times we'll talk about mitochondria and chloroplast. I think Karen talks a little bit more about the nuclear versus chloroplast genes today in her talk. In plants, we use this RBCL sequence, a MATK. I'm going to actually talk about a different sequence today, and I think Karen is as well. It's an ITS sequence. So these are all just different sequences. Again, these short barcode sequences that are conserved, but there is variation. And in fungi, we use the ITS as well. So this isn't an exhaustive list of the barcodes, but just to sort of give you some idea. All right, so to kind of walk through this process. So how do we do this? All right, so you start with something, right? You have to have some little scrap. One of the nice things about DNA barcoding is you don't need the whole organism, right? And what we've relied upon a lot, previously to identify is to look at the morphology of something, right? You know what a species is based on its morphology. Well, that's nice if you can have all of the informative parts, right? But sometimes you don't have all the informative parts. I work a lot on insects, and generally the informative parts of an insect are its genitalia. You can laugh, it's okay. So, like fruit flies, when we work on fruit flies all kind of look the same. Even when you know what you're looking at, they really all kind of look the same. But you can identify them a lot by what the males look like, their genitalia. So that's not always preserved, though, right? So if you don't have the genitalia, well, it's a fruit fly, right? I can't tell you what it is. But with DNA barcoding, that's not an issue for identification, right? We can identify off of things that are mangled beyond all recognition, right? That we don't have the whole sample for. So I'm using a feather here in this example, and I want to kind of put this out there. So DNA barcoding has actually been used to detect which kinds of birds are getting hit by planes. They can basically just take a sample of some of the feathers that come off of a plane that's been involved in a bird strike incident and be able to identify which species of birds are being hit by these planes. So actually, feather's a really good example. You're not always able to identify the species by just the feather sample itself, but using this process of DNA barcoding, we can. So we can start with something like the sample right here. We can extract the DNA pretty quickly. We're pretty good at this. There's kits, they're publicly available. It's pretty easy to do. Extract out the DNA, we keep it in a little tube. We amplify it at one of our primers. So this is that CO1 primer I was talking about. And we can amplify and make lots using a PCR machine. Sorry, that really important part of technology, right? Little PCR machine. We put the primer, we put some other stuff in it. And we do a PCR, polymerase chain reaction, right? And PCR lets us make lots and lots of copies of that short little bit of DNA, right? So using the primer and the DNA that we've extracted, we've now got lots and lots of copies of that CO1 locus that's in that feather. And taking that lots and lots of copies, we can then sequence it. So this is a little bit of an old-fashioned sequencer. It's bigger. I'll talk about some of the technology here in just a minute. But this is actually very close to what I used when I did my PhD. So the really big floor model. And when it comes back is we get all the A's, the T's, the C's, the G's. We get it generally in a pretty nice computer file. And then we take that information and we do something called BLAST. When I explain BLAST to my students, it's basically Google for DNA sequences, right? So I'm going to put the sequences in. I'm going to say, search for this. Tell me what this is, and it's going to blast it. It's an algorithm. It's a little bit more complicated than that. But so is Google, right? Googling, and it's a little more complicated than just saying just Google it. So you just blast it. And these are a couple different databases that are known for having barcodes. There's more, but NCBI GenBank is a really good one. It's all publicly available. BOLD is the Barcode of Life database, and they partner with a lot of different databases from around the world. So you run against the database, and then you can identify the species, right? It's going to come back and say, you've got 100% match to this species or this genus, if that is in the database. We'll talk about that in a minute. Or you've got a 97% match to something in the database, or 90, so maybe not exactly it, but really close. So you might need to do a little bit of extra footwork, and we can talk about that a little bit as well. So thinking about these databases, so where do the sequences in this database come from? How do we know what all of these are? Well, they come from museums a lot of times. They come from herbariums. They come from places where taxonomists have spent a lot of time collecting and identifying things the old-fashioned way, right? So how many petioles does that flower have, right? All of the really basic, well, not basic, the really complicated morphology stuff in a lot of cases. And so what they're doing is they're going through and they're taking a sample from that known specimen, and they're amplifying those barcode regions, right? And then they're depositing that information into those databases so that when you just have the DNA sequence, you can kind of back reference to those organisms. And a lot of people are spending a lot of time doing this. In BOLD, the iBOLD database, there are close to 5 million barcode sequences that are in and verified and documented, right? A lot of animals, a lot of plants, a lot of fungi. And even more if we include other databases. I think GenBank has a little bit more, a couple more. But we still have a long way to go. By no means is this an exhaustive list of everything in the world. There's a lot of organisms. So there's a lot of push to know more, right? To have more barcodes, to have more of this information. And as sequencing, we'll talk about this in just a minute, becomes cheaper and easier, we can start to make these barcodes a little bit, these databases a lot more full, I guess, to provide us with a better picture of what's really going on. But I put these up there because I think they're kind of interesting, these barcoding initiatives. There's some that are really location specific. The NYC Urban Barcode Project was a great one. It was actually high school kids going out and doing a lot of barcoding. You might have seen it in the news. They did some barcode sampling of the subway in New York. Where they kind of swabbed some of the poles, and I think it was kind of a joke because I think platypus DNA had come back. It was a really bad match. It wasn't the best of science, but it made a lot of people go, what was a platypus doing on the New York City subway system? And I don't know. I don't have the answer to that. No, but it was an interesting project to sort of get kids interested in science, but then to also contribute to this database. There's a lot of, and again, there's also really specific ones. The Norwegian Barcode of Life. There's a German Barcode of Life with a real emphasis on location. Really thinking about gathering as much as they can from a certain geographic location. But then there's also different consortiums that are surrounded, with amphibians or reptiles, right? Really trying to understand amphibians and reptiles. And a lot of this information that's in this database goes beyond just the barcode region. So I do a lot of arthropod genetics. That's kind of where I came in as an evolutionary geneticist. And we have a goal of 1,000 arthropod transcriptomes, which is a lot of sequencing. That's a goal. So there's a 1 IKTE effort, so 1,000 transcriptomes in I think the next year or two is the goal. So a lot of genetic information, sort of trying to fill in these gaps that we have in our information, and it's coming by really quick. There's even some, I like this one, the National Cockroach Project, to identify variation in cockroaches, looking at it, and this is another one that's done sort of as a learning tool, but is really providing as a basic science tool, but still really providing information for us as well. So if you see a lot of cockroaches, this is a citizen science project where people actually send in their cockroaches. I'm really glad I don't work on that one. I imagine they get a lot of really, like you stuff it in an envelope and mail it off. It's got to be pretty rough. I've had people send me like spiders before. And they're like, what is this? I'm like, I don't know, you stepped on it. It's a really dead spider, I think. All right, so now we've kind of gone through the DNA barcoding, and that's to go from a single sample to figure out what species that single sample came from. Another term that we're going to talk a lot today about is DNA metabarcoding, and this is where we're starting with a mixed DNA sample. So what I'm using up here is the example is pollen samples. So there's a lot of different, you can see the different colors and the different shapes. There's a lot of different species represented within this single sample, right? But we can use a lot of the same ideas and a lot of these same principles to identify all of the different species that are in there, or a lot of the different species that are in there. And that's because we have something called high throughput DNA sequencing. This allows us to look at lots and lots of different sequences. You get a big file that returns it. So again, a lot of the same procedures. We're extracting the DNA. We're still amplifying it sometimes now we don't, sometimes we don't have to rely on the primers, but we're amplifying it with this primer sequence. We're getting lots and lots of copies of the DNA. We're still going through sequencing. Imagine that this is a slightly different sequencer, but the same idea, right? They're just smaller and faster nowadays. And we get a lot of different sequences. So from a single sample, you might get 40,000 reads, right? It's about right? It's probably higher now. Since like last week, it probably got, now we'll give you 100,000 reads. But, around 40,000 individual reads of all of the different sequences that come in. We still use the same databases. We use some it's a little bit more technical, so we do some bioinformatic pipeline work, some programming. We were just talking about that before of, oh crap, it's written in Python. But anyway, we can sort of figure it out. People are pretty good at it. And then we can create a list of all of the different species that are represented in that mixed DNA sample. So that's DNA metabarcoding using high throughput DNA sequencing. I put this in here because it's one of my favorite examples. It doesn't quite use. It uses Galaxy. This is a little bit different with their metagenomics. They use metagenomics, but sort of really the same idea. What they did was they were interested in sampling the biodiversity in a state park, right? And you can imagine there's some really big state parks out there. So how do you do it? How do you do it effectively? And I think this started as a drinking grad student joke, but they decided what they would do was that they would drive through the park. And then when they got through to the other end of the park, they would just take a swab off the windshield and see how many bugs they'd hit. This is literally the sampling, if you read the paper. So they took samples off the windshield, they took samples off the bumper. They sterilized everything before they drove. They drove through, they took the swabs at the end, and then they used metagenomics to identify the species that they'd hit with their car on the way through the park. And it worked, right? Surprise. This, and I know James Taylor, actually you do too, he's at Emory. Anyway, who's on the paper, not the singer. But yeah, so this is kind of one of my favorite sampling technologies, I love it. All right, so going back to this idea of sequencing. So sequencing is getting faster and smaller and cheaper, right? So like everything, it's getting faster. So this is a really big, slightly outdated, I'm trying to remember what brand this is. I'm having a blank. Sorry, up in the upper left, this really big floor model that's person standing next to it. We now have Illumina, that's the one over on the right. It's a desktop, sits on the desktop. We do a lot of Illumina sequencing now. It's pretty quick. It's getting to about, I don't know, depending on if you're sending it out or somebody else is doing it. For some of those mixed DNA samples, you're looking at about $40 a sample for some of the sequencing to come back. And again, you're getting about 40,000 reads. And then sort of the new exciting thing is this is a mini ion sequencer. You can see that it hooks up to a USB to your computer. So it's very tiny. So we're moving into this idea of having handhelds. This was kind of a science fiction, Isaac Asimov might have written something idea just a couple years ago, and now we're really starting to take this seriously as a way of sequencing. It's not perfect. There's a lot of the technology that's a little bit, that still needs to get worked out, but we're really moving to smaller and faster and cheaper. And the mini ion, they're thinking about putting it out into the field to, especially for disease outbreaks, right? So it's pretty good at sequencing viral and bacterial genomes pretty quickly. So one of the reasons that it kind of came out a little bit early was to think about Ebola, right? So if you could identify something really quickly out in the field, you didn't have to send it off, that it would come back. So that was sort of one of the big pushes to get that. All right, and the sequencing, the actual sequencing tech, I'm not going to go through the slide, don't worry. Don't panic. All right, so the technology for how to do the sequencing, how to do it faster, cheaper, and more efficiently, these are just some of the methods that that people use and that are you'll hear things like, well, we don't talk about pyrosequencing too much anymore, but that was sort of one of the things in 454 sequencing. The little mini guy that I showed you, the mini ion, uses a nanopore technology kind of thing, right? So really thinking about how can we more efficiently, with less error rate, do a lot of sequencing, and that's something, PacBio is kind of the new thing that we're thinking about for genome sequencing. There's a lot of different technologies out there. There's a lot of different pros and cons to them, but the basic take-home message is we're becoming faster, we can do more sequences, and it's becoming more affordable. I'm just going to put this in here because Piper's going to talk about it a little bit. We can even start to think, because technology is catching up and we've got better applications and it's faster and all this, we can move beyond just barcodes and think about looking at the entire genomic sequence, right? So this is something she's going to talk about, RAD-seq. We can start to use something called next generation sequencing. So to sequence and identify populations, right? We can start to use the idea of looking across the entire genome, or even just targeted bits of the genome that aren't the barcode, to look where there's variation and where there's not variation. And this can help us identify individuals within a population, right? So we're going to talk about some of that with fishing and logging. This stuff becomes really important, thinking about RAD-seq and other ways of doing population genomics, really, and moving beyond just looking at one particular gene sequence. All right. So let me talk about some of the stuff that I've done, just really briefly to wrap up. So this is work that I did with an undergraduate of mine a couple summers ago in kind of a small undergraduate project. So two people with a small budget, nothing too big, thinking about how we can use pollen as a forensic tool. And I'm from Atlanta, Georgia, and it really does look like that. We don't have winter, we have pollen season. It's not during winter, but we just don't really have winter. And everything turns yellow, your car turns yellow, it rains, the streets turn yellow, they run yellow with pollen. It's mostly pine pollen. Pine pollen's everywhere. I don't have allergies, my husband does, but it's pretty miserable for me too. But yeah, so pollen is sort of everywhere. And we can really think about pollen as being a forensic tool, right? And people have thought about pollen as a forensic tool for a really long time, trying to think about where an object has been. You can look and see what type of pollen is associated with it and go, oh, well, that plant goes here, and so that, that person's probably been there. Traditionally, the way that that's been done is looking at pollen. So if you actually look at pollen, it's not just a homogeneous yellow thing. Pollen comes actually in all different kinds of colors. It's really pretty to look at. You see lots and lots of variation. And if you know what you're doing, you can tell what species this came from, or at least what genus it came from. Can't always do the species identification, but you can start to tell which plant is which. And this has sort of been done largely through lots of looking down microscopes. I'm not trained in this. I will. That's my caveat. Right? So visually identifying, going through a sample and saying, all right, this comes from this plant, this comes from that plant, right? But people do this. They've been working on ways to use this to do this a little bit more high throughput using computer learning, having computers look at the slides and identify what species of pollen that came from, and that's still in some form of development. But as you might guess, we'd like to look a little bit closer, right? So thinking about actually using DNA and these barcoding methods that I just talked about to do the identification work for us. So again, this is me and my undergraduate student. She's now graduated. She's going to medical school. She just graduated this year. So I'm really proud of her. We collected surface soil samples from 4 different locations. We used the pinch collection forensic method where we just sort of went around and did a pinching sampling of the topsoil in an area. We picked 2 around here, one in El Paso, just sort of north of campus, and one of the washouts up there. We also went out to the India Mountain Research Station, which is UTEP's research station out near Van Horn, and we did, again, some more pinch sampling. During the course of the summer, went to Raleigh for a conference, and so I did some pinch sampling in an empty vacant lot over there, and then I live in Decatur, Georgia, so I did it in my backyard too, because why go far, which is right outside of Atlanta. All right, so we used meta-DNA barcoding this time. This time we started with a soil sample that looked very much like this, right? Little bag, put it in a little baggie, and we did we did our DNA extraction from that using a standard soil extraction DNA kit, and did a little bit of modification. We shook it a little bit harder and a little bit longer than you normally would to really crack open those pollen granules. And again, sort of this last step of running it through the databases and coming back, we used an ITS pipeline that's not here, but that's okay. So I will say we identified most plants to the genus level, and that's because most species were missing from GenBank. It looked like there were a couple that were 100% match, came right back, and I'll tell you what those were, and they make sense. So in El Paso, I think you might know this one. It's the monkey comb plant. About 100% of that sample was that, and it was a dead-on species match. That was what was in there. Not too surprising, right? In Indio Mountain, again, really rough comparatively. Like Georgia's really green, so when I come out to El Paso, I'm like, wow, not a lot of plants, right? But there are. Indio Mountain, we got a hit to tomatoes, which is a kind of a head scratcher because there are no tomatoes during the heat of the summer out in Indio Mountain. But it's basically to the genus of nightshade, right? And there's a silver nightshade. You guys probably know this plant, right? You see it around El Paso if you're in El Paso. And so I think 97% of the sample was this plant here. Then Georgia, my backyard. Veronica is the pretty little blue flower. That was a genus match to Veronica, but I know it's there because I planted it. So it worked out. The one on the far right with the pretty purple seeds is something called pokeweed. If you're a southerner, you know pokeweed. That was a dead-on species match, and I will attest to the fact that it's in my backyard because I've been fighting it for years. And then the one on the left actually didn't have a species hit, but it's an invasive. So it hit to a genus that comes out of Japan, and it's the Japanese chaffweed. And so that was sort of the third. So those 3 really made it up. Raleigh in the vacant lot. We got a grass. It looked like Bermuda. Again, that was a hit to the genus, which is Bermuda grass. Probably a good bet. Something called lamb's tongue, which is really rampant in vacant lots, and a flowering mustard. And again, we couldn't identify to the species level on that, but pretty sure that all makes sense as far as geographically what's there. One thing I'd like to point out is that all 4 samples we got pollen information, not any overlap. Right? So they all look different from each other. Now, there are some missing things, right? But we can start to think, well, that's what this area looks like. That's what that area looks like, right? So it looks very different from each other. The species level thing is something to note, right? You can't always identify to the species level just because that's some of the gaps that we have in our database right now. So it's one of the limitations. But that's also true often of pollen visual identification. So it's sort of in both of those. All right. So I'm going to be wrapping up kind of quickly. So just to review, we can use this DNA barcoding to think about questions of provenance, right? So one of the things that I've done in my lab as well is look to see what pollen's in honey, right? So there's pollen in honey. You can get the DNA from the pollen out of the honey, and you can start to think about where does Where did that honey come from? Again, people visually have done it, but we can really start thinking about using DNA metabarcoding and high throughput sequencing to answer these questions as well in the same methodology. And like I said, so Piper's going to be talking about some case studies, thinking about how we identify where populations come from. Oh, and I do want to say, Karen's going to talk, she's our method. She's got some really fascinating methods that she's been using really recently, new cutting edge stuff that's I was really excited. I'm really excited about thinking about how we do pollen DNA barcoding. And then I wanted to wrap up because I think this is, I'm an educator, and I think this is a really great example of DNA barcoding, and it was Sushi Gate. So there were these students in New York. They wanted to know if the sushi that they were eating at the restaurant was actually the type of fish that was advertised, right? Was there tuna actually tuna? And it turned out that it's not always the case, right? So, what was being sold as white albacore tuna when they did the DNA ID is actually tilapia, a lot cheaper, right? And so really kind of a basic science application that I think is a really interesting one. Again, this was done by these two students up here as part of their class. So really accessible technology in some ways. And so these are some of the barcodes that came out of that work, showing the actual using the barcodes to identify those species. And so with that, I'll sort of wrap up. I think my time's up, and we can open up the floor to questions. I thank you very much for your attention, and I'm really interested to hear what we have going on. So thank you.